Thank you, Jekyll. I wanted to just welcome all of you for attending the MAPP rounds. We're very excited to have Dr. Lea Grinberg with us. And so I'll leave Cynthia Agüero to talk a bit more about her background.
Hello, everyone. It is my great pleasure to introduce you Dr. Lea Grinberg, MD, PhD. She received her MD and PhD in Sao Paulo, Brazil. Currently, she is an associate professor of neurology and pathology at UCSF. Her research focuses on neurodegenerative disease with a special emphasis on early disease stages, selective vulnerability, and pathological heterogeneity. She is a member of the governing board of the Brazilian Biobank and Aging Studies. She is part of the executive board of the Global Brain Health Institute and the medical and scientific advisor group for the Alzheimer's Association. So thank you very much, Dr. Lea, for joining us today. Please, everyone, if you have questions during the talk, please write the questions in the chat, and we will read that questions at the end. Thank you very much, Dr. Lea, and thank you very much.
Thank you very much. We are just commiserating that I would rather do it in person, but after I've heard that it's snowing there in Boston, I'm not so sad that I'm here in sunny San Francisco. It's not raining for months here, but this is another story. It's really a pleasure to be here today. I would like to thank you very much for being here with me, and of course, special thanks for Yakel for inviting me. It's really an honor. And today I will share my screen now. Let's see if it works fine. Can you see my screen?
Yes.
And I'm not sure if you are seeing the right screen. Are you seeing the one that has two?
We're seeing the presenter's notes.
The presenter's note. Okay.
Oh, so the presenter's view. Yeah.
Yeah, let me try to do a new show. Oh, okay. All right. I think it will work now. How about now? No?
Perfect. Yes. Now we can see the slides.
Wonderful. So what I would like to share with you a little bit, it's a little bit of my trajectory on research in Alzheimer's disease in the past years and how it evolved from, a single observation when I was on my PhD to a lot of research I'm doing with collaborators all over the world nowadays. And it's all about rewriting the initial stages of Alzheimer's disease. As probably many of you know, Alzheimer's disease is a growing epidemic. Now that we hear about COVID, I think we have a different definition of epidemic, but still Alzheimer's disease is there and numbers are growing. It was estimating that in 2015, more than 20 million people worldwide had it, which means 1 in 8 people age over 55. And the costs of dementia are very high because someone that develops dementia can be in this condition for even decades and always declining and always in need of more help and all the other care that's necessary for someone who is developing dementia. And if you look at numbers, you can see that dementia, or they call here Alzheimer's disease, but probably include most of them. It's the only one of the 10 most common causes of death in the US, in which numbers are increasing in the past years. And we don't have treatment yet. A lot of promises, some failures, but it's kind of unknown when we will be able to really treat the disease. So Alzheimer's disease, as other neurodegenerative conditions, have neuronal loss and have accumulation of abnormal proteins. But a very important feature that sometimes is neglected in some studies is that neurodegenerative diseases spread in a stereotypical way. What does it mean? It means that each one of these diseases or classes of diseases, usually, most of the cases, will develop first in certain areas of the brain, and then it will spread in a way that we can predict, usually through neuronal networks. So because of this, it makes kind of sense to consider that if we know where the disease starts, probably we can design tools and even treatments that will be working in these most vulnerable areas. So it seems like a no-brainer. But when we think even in Alzheimer's disease, and we think that we want to understand where in the brain it developed first, it's not so easy. And one of the reasons for this is because the clinical symptoms, or at least what we call disease-defined clinical symptoms, just show up years after the pathological process starts in the brain. So we need other tools to understand where the disease starts. So when we are trying to do it, we have to ask what is the best marker of progression. So in the case of AD, many clinical pathological studies showed us that the best marker of disease progression is neuronal and synaptic loss. And I brought this nice illustration here. This is a cut of the human brain. Here is the entorhinal cortex. Probably you've heard about this cortex, very early vulnerable to Alzheimer's disease, especially this pre-alpha or layer 2 cells over here. And this is a case, it's a normal patient. You can see the islands of cells. And this is Alzheimer's disease where you cannot see anymore. The problem is it's very difficult to estimate neuronal loss, especially when this is very mild. And one of the reasons for this, it's not only lack of resolution to do it in vivo, but each one of us, we have a different numbers of neurons to start with. So we don't know what is the baseline. And when we don't know what is the baseline, it's difficult to measure when there is loss, especially if the loss is mild. So we use the best second thing. And the best second thing in Alzheimer's disease is the burden of neurofibrillary tangle, of tau changes. And to illustrate this, I brought just one of the work that show this clearly for us. This is the cohort from the São Paulo Brain Bank that has, at that case, 1,200 cases, and it's a population base. So what we did here, and Claudia Suemoto led this study, was to give a score to each one of the changes that we see in the brain, and then doing some statistics for creating this score of what affects more cognition. And as you can see here for this BRAAK NFT stage, that means the amount or the distribution of tau changes in the brain. When you have the later stages, you get 7 points, which is many more than any other conditions we see there. And if you pay close attention, we don't even see amyloid depositions there, because in our studies and also in our studies, it seems that there is a very poor correlation between amyloid deposition and clinical outcomes. So when we think again on tau, that seems to be the most important marker, at least a marker, I'm not saying a cause, so please let's make a distinction about it, of disease progression, we automatically think on the BRAAK and BRAAK staging. So this was a stage that was proposed by this couple from Germany in 1991, using exactly this concept of stereotypical spread. So what they found out is that examining 2,000 brains, they will never find tau deposits in areas that now we know are later stage without finding this same tau in early stages. And for this specific staging, they look at the cortex. So they define that the entorhinal cortex, the same one I showed before, it's the first one to accumulate these AD tau changes. And then as the disease progresses, you start having more involvement of limbic and paralimbic areas. And finally, you get to associate cortex. And the last stage, you get to primary cortices. This is very reproducible, and we use in all our studies. The point is the cortex really the first region to accumulate what we call AD tau in form of tangles or pre-tangles? So as I told you, I start my career in Brazil, and over there in Brazil, autopsy is still mandatory. So we have a very well organized autopsy center at the University of São Paulo that provides for the city and conducts about 15,000 autopsies per year, which is unheard in other places. So back in 2003, we decided to leverage this resource and build a research study on aging using this autopsy service. Of course, the families have to enroll, but we could build this study. So because it's population-based, as opposed to most of the cohorts that come from dementia centers, so people are late stage, we started to get a lot of people over 50 that come to autopsies for whatever cause, and they didn't have cognitive decline. But when we were examining their brains, we started to detect these early stages of neurodegenerative conditions that we talked before, because these diseases, they spread in the brain for many years before we have clinical symptoms. And indeed, if you look at the cohort in Brazil, the CDR0 means controls, you see that the majority of the cases, they are in this category. And most of them, like 75%, they have a normal neuropathological diagnosis, because we either find no change or we found very little change. But as you can see here, 25% of them already meet diagnosis criteria for some neurodegenerative condition, even if they are controls. So these are the groups that we were very interested in looking at, because they will show us where maybe the disease starts or where the first and initial symptoms. And in fact, looking at these brains, we started to see something very interesting, that cases that didn't have any pathology in the cortex, so remember, cortex, entorhinal cortex, first stages of tau deposition, they would already have very similar tau depositions in subcortical areas, more specific where I put this dot here, which is the dorsal raphe nucleus that produce 80% of the serotonin we have in the brain. So, you know, description of tau changes in subcortical nuclei were not something new. I mean, we have plenty of these descriptions for the 70s and 80s, but it was not very clear at that point how early these changes would happen in the brain. So I was lucky at that time, because I was going for a postdoc in Germany. And one of the former mentees of my mentor, Udo Rupp was the mentee, he was also interested in this field. And he had published a paper a few years back showing that the same normal dorsal raphe that I saw changes seem to have progressive changes in Alzheimer's disease, and seems to start very early. But again, he could not determine if it was before or after cortical involvement. But I could, because I had a lot of cases. So this is exactly what I did. And in this study published in 2009 with 120 cases and 40 of them without cortical pathology, we could demonstrate that every case that had pathology or tau pathology in the cortex already had in the dorsal raphe. But we still had many cases with pathology in the dorsal raphe, but didn't have anything in the cortex. So, you know, we linked the dots, and we used kind of the same method that BRAAK and BRAAK used in 1991. And we proposed that this subcortical pathology maybe would be preceding cortical pathology in Alzheimer's disease. And this was followed up by some other papers. And in fact, BRAAK and his group re-examined their cohort to look at more closely to subcortical pathology. And they also detected in 2011 that in that cohort, subcortical pathology came before cortical pathology. In that case, they were looking at the locus coeruleus, which produced noradrenaline, another one of the subcortical structures. And we also continue to look into this, and we published this paper together with Udo Rupp in 2016, showing that not only the dorsal raphe and the locus coeruleus, but about 15 subcortical structures, including all the cholinergic nuclei, they start accumulating AD tau before the cortex. So far, so good. But the point is, and this is a question we've heard a lot, they said, okay, you are detecting these early changes in subcortical nuclei, but the subcortical nuclei, they represent almost nothing compared to the number of neurons we have in the brain. We have 88 billion neurons in the brain. If you take all the subcortical neurons together, they represent less than 1%. We are not even seeing, clinical changes there. Does it matter, or you are just in an exercise of futility trying to find details that are not important. And this was very fair questions, and we decided to explore it more. And the first thing we have to do was to find the right method. Without the right method, we cannot do it. And we decided to use this method developed by Helmut Heisen, in which we take whole brain structures. We'll put in something called celloidine instead of paraffin, which is a media that has been used at the beginning of the 20th century and Helmut kind of rescue. And celloidine is very good to produce fixed sections. And fixed sections are very good for doing counting with a method called stereology that provides unbiased results. And this is exactly what we did. And stereology is very important because when we just count cells looking at one slide through the area, we might be hitting, so many different combinations that the results are really not reliable. And the other thing that we leverage, and it was to our advantage, is that because of this brain bank in Brazil, we had cases throughout BRAAK stages. So we could really model the disease progression. And this is exactly what we did. So we went to answer different questions. So the first question we wanted to ask was if the subcortical tau burden that we were seeing there was really pathological or just an epiphenomenon of aging, and we should not really be wasting our energy on this. So the first thing we try to understand is if people that age, but they don't have accumulation of neurodegenerative changes, would they have changes in these structures? And Panos Theophilus, who is assistant professor in my lab now, he looked at this in the locus coeruleus, and we concluded that age by itself did not cause changes in the neuronal population or in the volume of the locus coeruleus. And Sarah Dunlop, who was in my lab and now is a PhD student at Northwestern, she looked at the same thing with the dorsal-raphe nucleus and also no changes. And we even look at the substantia nigra that also can be affected early in Alzheimer's disease. And this was Ana Teresa Alho. And again, we found no changes, maybe a slight change in volume with aging, but nothing else. So what it told us is that if in our subsequent studies, we will find changes related to accumulation of abnormal proteins, probably these changes were not related to aging, but related to whatever process was going in together with the deposition of proteins. So this is exactly what we wanted to study. So to study this, we wanted to know if the subcortical tau depositions will increase in number and severity in the same way that cortical pathology does. Because again, neurodegenerative disease, they have to be progressive. If you just have a deposit for whatever reason, maybe you had some inflammation or whatever thing, and then it doesn't progress, it doesn't really fit the bill for neurodegenerative diseases. So this is the same study I showed from 2016. And you don't need to be a pathologist or a neuroanatomist to recognize that in several of the subcortical nuclei, even in BRAAK 0, when there is no changes in the cortex, you already can see some tau pathology here. That of course, by the time you have late stage disease, they grow by a lot. So certainly this pathology was progressive, but we wanted to measure this. And Alex Ehrenberg, that was in my lab at that time, decided to look into this, and we did quantitative assessments of the locus coeruleus and dorsal-raphe, and we could show that at BRAAK stage 0, again, no pathology in the cortex, already 8% of the neurons in the locus coeruleus had what we call AD tau depositions, and by the time it moved to BRAAK 1, when this pathology was starting to happen in the cortex, this number doubled to 16%. Showing us or already pointing to us that there is indeed progressive activity in subcortical nuclei, and they happen very early in the disease stage. So then there was this question, okay, tau deposit in the subcortical nuclei and it progresses, but does it cause any harm? Or it's just, very benign, and again, a futile exercise to bother looking into this. And this Panos came to the rescue again, And what he did was to take these progressive cases of Alzheimer's disease, and he looked again at the locus coeruleus and the dorsal raphe for signs of cell distress. So he looked for neurons that had active caspase 6. Caspase 6 not only participates in apoptosis, but also truncates tau to a very toxic tau form. And what he could demonstrate is that, in fact, there is a progression of the number of neurons that have this active caspase 6, and usually it overlaps with the neurons that will have accumulation of tau AD, also demonstrating that this accumulation of tau AD doesn't seem to be very benign in this nuclei. So it's something for us to be paying attention. And indeed, when we went to count neurons in progressive stages of the disease, we could identify that in the locus coeruleus, we start having changes in volume of about 8% per BRAAK increment, starting from the BRAAK 0 to BRAAK 1. So again, even before we have pathology in the cortex, this nucleus already loses about 8% of its volume. And also early in the disease, we start seeing neuronal loss. And actually by BRAAK stage 6, the locus coeruleus loses about 80% of its neurons, which is a lot. Interesting, we didn't see the same changes in the dorsal raphe. That opened questions about the selective vulnerability and how much the same tau can be toxic for some neuronal populations, but not for other neuronal populations. And I will have the opportunity to talk a little bit more about it. So when we think about selective vulnerability, we usually think if a certain neuronal population is prone to develop, for instance, AD tau or alpha-synuclein or not. And we tend to consider that if they will develop these abnormal proteins, they will probably die. But if they don't develop these proteins, they are protected. But actually, in our studies, and we could see it very well with subcortical nuclei, we detected a different layer of selective vulnerability. So the stage number 1 is the neuron will accumulate abnormal protein. And the stage number 2 is what happens once this neuron accumulates this abnormal protein. And the way we studied this was looking at different tauopathies. So, disease that accumulate tau, but different from AD tau in several of these subcortical nuclei. And what we found out is that, for instance, in the locus coeruleus, I will just use this example. Although we see protein accumulations in Alzheimer's disease, progressive supranuclear palsy, and cortical basal degeneration, the last 2 or 4 repeat tauopathies, the neurons only die, you see here in orange, in Alzheimer's disease, but not in PSP and CBD. So we say, oh, so it means that the tau from PSP and CBD is not as toxic as AD tau. Well, we don't think so, because then if you look at the substantia nigra that also accumulates tau in these 3 conditions, the neurons only die in CBD and PSP, but not in AD. So study subcortical pathology also give us a glimpse about this idea of selective vulnerability, in which is not only a matter of accumulating some sort of abnormal protein, but is the combination of this abnormal protein with the neuron that's there. And this is very important, I would say, for modeling studies, experimental studies, in which we try to consider neurons as a more homogeneous thing. So finally, we wanted to ask the most important question. Does it have any clinical impact? I mean, what is subcortical accumulation of tau causes? We knew it was not directly memory loss because it started to happen before memory loss. But then we know from the literature and epidemiological studies that people that will develop dementia, they tend to have higher rates of some neuropsychological symptoms before they go to develop dementia. And this can be, in a way, measured by several questionnaires. The one we use in Brazil is called Neuropsychiatric Inventory, NPI. which is very well used everywhere. So what we did was the following. We took our 1,200 patients. We eliminated everybody that had some pathology in the brain that was not Alzheimer's disease, because it will be a confounder. And then we ended up with 455, which is a lot. So out of those, 130 didn't have cortical pathology, BRAAK 0. And 200, they have very little cortical pathology, BRAAK stage 1 and 2. So what we found out is that if you compare this BRAAK 0 to BRAAK 1, they already have a much higher odds ratio of several of these neuropsychiatric symptoms, for instance, as agitation, and but also appetite dysfunction, anxiety, and a very interesting for us was the sleep disturbances. And in fact, some studies come to suggest that sleep disruption is a risk factor of Alzheimer's disease, and or at least of dementia. So we wanted to ask if it's really a risk factor or it's already a consequence of the pathological deposition of abnormal proteins that we see in Alzheimer's disease. When we think in sleep control in the brain, we talk about nuclei that control awakening and nuclei that control different kinds of sleep. So to be simplistic here, I'm showing this schematic, showing some of the nuclei that control awakening. So for us to be awake, this is an active process by the ascending arousal system. And the locus coeruleus that we talk a lot today, together with two other nuclei in the hypothalamy. One it's called tuberomammillary nucleus. It produces histamine. So for everyone that ever used Benadryl and felt drowsy, it's because of the histamine here in the tuberomammillary nucleus. And this lateral hypothalamic area that has a population of neurons that produce hypocretin or orexin. So we wanted to try to understand if excessive daytime sleeping that we see a lot in patients with Alzheimer's disease, even before the onset of memory loss, could have anything to do with the generation of these three nuclei. And why we hypothesize this, because in our previous studies, these three nuclei were part of one of these 15 that accumulate tau very early on before cortical tau. So the hypothesis, it's kind of the following. So when we think in sleep-awake patterns, we have wake neurons. They are awake during the day. And when we need to sleep, we have this flip-flop switch model that make them kind of switch off. And then the sleep neurons start to become active. So our hypothesis was that in Alzheimer's disease, we'll have a lot of loss of these awake neurons, but not of the sleep neurons so much, at least in early stages of the disease. And this is what we'll see people that could not stay awake. But also to create also a disease control paradigm to our study, we include people with PSP. So PSP is this for repeated tauopathy that I mentioned before. And patients with PSP, they cannot sleep. So it's the opposite of Alzheimer's disease. They only sleep 4 hours each night. It's very devastating. And this is work from Christine Walsh and Tom Nyland at UCSF. So going back to our model, we hypothesized that in PSP, what we'll see was a preservation of wake neurons, but then a decrease in sleep neurons. And Joseph O and Rana Ezer took the leadership in this study. We used stereology again. And the first thing they saw was that when you look at tau deposition in locus coeruleus, this orexin neuronal population, and the tuberomammillary nucleus, in all three conditions I will show here, Alzheimer's disease, cortical basal degeneration, and progressive supranuclear palsy, we have a lot of tau deposition, and in controls, of course, we don't have tau deposition. So it seems, you know, that in these three conditions, these three nuclei are vulnerable. But then when we look at the number of neurons that are out there, and comparing here with controls, this very dark blue, we can see that neurons die in Alzheimer's disease. In CBD and in PSP, we didn't detect neuronal loss in the hypothalamy, and we detect mild neuronal loss in the locus coeruleus, incomparable to what we saw to Alzheimer's disease. So, and then we went ahead and we said, okay, maybe the neurons don't die, but they stop to produce the neurotransmitter they have to produce. And then here we found more effects on PSP and CBD. It's not that tau is completely harmless, but certainly the devastation is way worse in Alzheimer's disease. So because of this, we wrote this paper last year that got a lot of attention, showing that there is a possibility that primary tau degeneration of this awake nuclei is responsible for excessive daytime sleeping in Alzheimer's disease. And why this is important? Because the field, based on data, again, had the hypothesis before that excessive daytime sleepiness in Alzheimer's disease was probably a consequence of a bad sleep night. People with Alzheimer's disease, they have fragmented sleep. So it was always like a reaction of sleeping bad and having to sleep during the day. And with these findings, we are proposing that, no, this excessive daytime sleepiness probably is also primary. And if we are to develop one day effective treatment for tau pathology, probably patients, even before they have memory loss, they can benefit clinically by having better sleep night, treating these changes in tau and subcortical nuclei. And also this study had another very important clinical consequence. So we talk about PSP and how these people don't sleep. So based on this hypothesis that they don't sleep because the awake neurons are up and the sleep neurons degenerate. So the problem with them is that too much awakening. Tom Nyland again and Christine Walsh started this very exciting clinical trial that it's ongoing, trying to treat sleep problems in PSP instead with zolpidem, which is Ambien, the most common treatment for sleep that don't work with them. It's by shutting down the awake system with drugs against orexin or for orexin receptor. So we should have first results at the beginning of the year. It was a little bit delayed because of COVID, but this is moving forward. So because we are very interested in clinical consequences, we said, okay, we got these interesting findings on the awake nuclei degenerating in Alzheimer's disease. Can we link them together with actual sleep metrics? And we could do it, or we are on the way of doing it, by having a good number of research participants here at the Memory and Aging Center in San Francisco that during life, they receive polysomnography, and then they donated their brains. So we could do correlations of neuropathological metrics, quantitative neuropathological metrics with quantitative sleep metrics. And in our preliminary results, what we found is that indeed there is a very striking correlation with changes in orexin neurons and histaminergic neurons with these sleep metrics. But for the locus coeruleus, we are finding some paradoxical results. It's kind of going the opposite direction. So we always learn something new. So what we are doing now, it's increasing the sample size and trying to look in some other directions to try to understand what are these results. We are also now looking at sleep promoting nuclei. And this is a very interesting nuclei called intermediate nucleus. It seems to be a hotspot for promoting what we call no REM sleep. And Mihovil Alarikov in my lab, he is looking at a very also exciting population of neurons that I bet that many of you never heard about, but super important, which is called melatonin-concentration hormone. So these neurons, they are located in the same position as the orexinergic neurons, but they don't overlap. And in animals, they control a lot of physiological functions, including appetite, sleep, but coordinating all this together. So we are looking at this population in AD and PSP, and apparently it's not affected as bad as the orexinergic neurons. I think it's good because probably if they will be affected too much, it's not really possible to live without this population. It's very important. Now Mihovil is trying to put all of this together and link the dots. Animal studies also support this idea that subcortical early pathology in Alzheimer's disease is deleterious. This is work from David Weinshaker with rats. Rats are interesting because they produce endogenous tau. And what they did here in this penicillin and APP tau rat model, first they found out that these rats, they accumulate tau in the locus coeruleus, despite they are amyloid rats, as early as they accumulate amyloid. And when you manipulate this locus coeruleus, you have a worsening in learning paradigm. So just showing in an experimental way that early affection of the locus coeruleus is problematic also for learning. There's also a series of work from many other groups, including from Michael Haneke, who was very pioneering on this, showing that if you manipulate the locus coeruleus in animal models for Alzheimer's disease, what happened is that the number of plaques in the cortex explode. And then when you rescue the locus coeruleus function, this number of plaques go lower. And it's the mechanism behind this has to do with neuroinflammation. So it seems that the locus coeruleus has a very important role in controlling neuroinflammation. Neuroinflammation now, it's really something coming up. It's very important in Alzheimer's disease. So this early involvement of subcortical nuclei not only seems to give clinical symptoms by itself, like the sleep or the mood, but also seems to accelerate cortical pathology in Alzheimer's disease. So another good reason to be looking into this. I have a few more minutes, and I just would like to talk briefly about, so what is the next step, what we are doing with this. So as pathologists, you know, we are very proud that we can detect early change, but it will only make a difference in the clinical setting for patients if we'll be able to translate what we can find under the microscope to the clinical setting. And the way to do it is to produce biomarkers. So in this, we decided to leverage this idea that locus coeruleus seems to be losing volume from very early stages of Alzheimer's disease, and maybe try to develop some biomarkers to detect these volumetric changes in the locus coeruleus using neuroimaging. So it's almost like, you know, trying to push a little bit the timeline of where we can detect changes in the brain before the onset of clinical problems. So the problem of doing that is, and I know that many of you in the audience do imaging of locus coeruleus, is that locus coeruleus is very small. So it's a problem for neuroimages because of resolution. And to make a comparison, I'm showing the volume of a hippocampus here in yellow and the locus coeruleus in red. So as you can see, it's super small. And this is how, you know, the points look like when you do imaging. You cannot see anything. The locus coeruleus was supposed to be here where I trace in pathology here. So, you know, with a team of researchers and friends from all over the world, we decided to take this challenge. So we said, well, maybe we can use high resolution histology and, you know, take MRIs of the same brain and then register them together and get to better ways to detect where the locus coeruleus is. And we started to do it with a 7 Tesla machine back in Brazil, and developed a lot of algorithms to deal with this registration, because registration of neuroimaging methods, it's not so easy. I think for people that work with PET and MRI, you can feel the pain. But when we talk about histology and neuroimaging, it's even worse, because we are always forced to slap the brain to find things when we extract the brain from the skull. And because of all the alcohols and other things we use, this is our end product. So it's like a puzzle to put it together. So we developed some methods for doing that. I won't have time to discuss more details on the methods, but certainly I can discuss it offline and send you the papers, and to come up with a very good registration algorithm that was created by Mariana Allegri in my lab that can really do great voxel-to-voxel registrations with pathology and MR. This contributed to efforts that are made by researchers all over the world trying to get better images for locus coeruleus. And I also want to make a shout out for Heidi Jacobs, who is a friend and colleague also working with locus coeruleus in cognition and Alzheimer's disease. And I'm showing her work really to illustrate what is, how powerful and impactful is to have good markers for this subcortical nucleus. So what Heidi showed here in this recent paper is that the locus coeruleus, it's very important for memory consolidation during awakening, maybe not alone, but with also other structures like the amygdala. But if we are able to measure in life locus coeruleus activity and locus coeruleus change of volume, probably we are in a much better condition to measure changes in the very early stages of Alzheimer's disease that will be important for monitoring progression and also for efficiency of clinical trials. So my takeaway of this today, and I hope I could convey my message even by Zoom. I can never see your face, so it's hard for me to calibrate the talks, but is that the nuclei of the neuromodulatory subcortical system, it's early vulnerable to Alzheimer's disease. It has an impact on clinical symptoms. It has an impact on disease progression, and it's understanding it better is a great opportunity for early detection and treatment. Finally, I would like to do some advertisement of the neuromodulatory subcortical system PIA, which is new with the start. So for all of you that are working on this or they are more curious to learn more about it, please join. We are planning a lot of activities, and for future steps, I mean, we'll continue to try to develop biomarkers and try to understand the molecular identity of these vulnerable neurons, and hopefully come up with an inform on better treatments, not only for the memory loss and disease progression, but all these neuropsychiatric symptoms that we see preceding memory changes, and it seems to be due to Alzheimer's disease substrate. Of course, I could never have done it by myself. I'm very grateful to my lab here at UCSF, the groups in Brazil, all the collaborations from all over the world. I'm really looking forward to meet all of you in person sometime soon, and of course, all the funding agencies. And thank you very much. Happy to answer questions if you have.
Yes, thank you very much, Lea, for that amazing presentation. We do have some questions here. The first one says, very nice talk, thank you. What is the percentage of the resilience individual in your cohort from the Sao Paulo, She is asking about the BRAAK 5 and 6, the individuals who were cognitively normal.
Oh, thank you. That's a great question. And this is a group that we are very excited in doing studies because they accumulate tau and amyloid, but it doesn't seem to have an effect on their cognition. So these cases are not so rare. Actually, this population studies show that about 20% of normal elderly that come to autopsy and have at least moderate burden of Alzheimer pathology, they don't have clinical decline. This is especially true in the oldest old. There seems to have a disconnect between plaques and tangles. And I would say that we probably have more than 50 cases. Oh, so wow. we have been using them for genomic studies. And we have a very nice paper that was published in Brain a few years ago, comparing these cases with symptomatic Alzheimer's disease, just to show that what really makes a difference is the neuronal loss. So these folks, they don't have neuronal loss despite accumulating all these proteins.
Do you have any hypothesis of why it's like that, or?
No, but we are trying to work on this. I think it has to do with, of course, the genetic components that these people have. And again, we know that education you know, it's protective against dementia. But now we are talking about the biology. It has nothing to do with this. So we're talking about people that develop a lot of plaques and tangles and the neurons don't die. So I think it has to do with the genetic composition.
Well, thank you very much. We do have another one. He says, amazing work as always. Has your group or anyone that you know look at the SCN in terms of tau or neuronal loss in the progression of Alzheimer's disease or dementia? I ask because sleep-awake dysregulation may lead to the circadian disruption. In more general, how can we separate the sleep issue from the circadian issue in AD? How
yeah.
can this method help answer these questions?
Thank you for this question. This is something that we have been asking ourselves for some years. The SCN, I think you mean the suprachiasmatic nucleus, which the clock in the brain that regulates the circadian rhythm. Yeah, we are looking into this. There is evidence in the literature that the suprachiasmatic nucleus shows some degeneration in Alzheimer's disease and also in PSP. In our own studies, we see tau accumulation in this nucleus, and Mihoville is studying this. So he is using a combination of these stereologies. So we are counting neurons, and not only counting neurons, but we are counting neurons based on their identity, because the suprachiasmatic nucleus have different neuronal populations, and they have different functions. And also we are starting to use very advanced methods like nanostring and the digital space profiling to understand this. If we will be able to distinguish what comes from you know, the suprachiasmatic nucleus in terms of the circadian regulation versus what happens in sleep promoting nuclei, it's to be seen. But we think we will, especially if we have solid quantitative data of the whole system, which we are working to have. But it will take some time. Thank you for the question.
Thank you. And we have one more. She said, excellent work. Thank you, Lea. Do you have idea why these subcortical nucleus are vulnerable to early tau?
There are many hypotheses for this. I don't think we have a definitive answer, but some of these hypotheses are that these nuclei, they are very big and they have very thin axons, but very long. So the metabolical, you know, stress on these nuclei, it's very high because they have to serve many, many neurons. So, you know, this stress over life may make them more vulnerable. There are some other hypotheses showing that some of these nuclei, they are located close to segments of the blood-brain barrier. They are more porous, and you know, the brain produces substance that it should get out. For instance, and they go through the area postrema, for those who are familiar with the brain-blood barrier. But also it makes these areas more vulnerable to receiving toxins from other places. And this could expose these neurons to these toxins and then develop disease. There are also other theories that has to do with the blood-brain barrier. For the locus coeruleus, for instance, the locus coeruleus innervates the vasculature. So, for instance, there are studies showing that people that get poisoned by mercury and they die acutely because of the high levels of mercury. If you examine the brain, the locus coeruleus is where you find mercury. So the locus coeruleus has this tendency of accumulate trash from outside earlier than other areas. So all of these theories are out there, but as I said, no answers yet. I have to say, you know, that the funding for looking at subcortical changes in Alzheimer's disease was not very favorable for many, many years. It's changing now. There is more interest. The data is more solid, showing evidence that we should be looking at that. But it's not that it has been funded as well as other areas of interest. So I hope it will, our knowledge will change in the future now that we have more funding.
Yeah, and just to finish, Heidi Jacobs says, thank you for the shout out of in the great talk.
Thank you very much.
Cynthia, there is another question in the QA.
Which one? Which one?
Not in the chat, but in the QA. You see
Oh,
the
oh, oh.
Yeah.
Yes, is one more. Wonderful walk and talk. I will have two questions. What are your thoughts on the noradrenergic hyperactivation preceding LC degeneration? That's the first one.
Yeah, that's a very interesting question. So despite the fact we know that the locus coeruleus is early affected and the neurons die, if we measure metabolites of locus coeruleus activity, in people with progressive stages of Alzheimer's disease, what we see is actually there is an increase of activity and not a decrease of activity. We are trying to understand why it happens. We know from RNA studies from Isidro Ferrer group in Spain that in fact you have increase of RNA expression in locus coeruleus in the early stages of the disease, so almost like a compensation. But for how long it lasts and what exactly is going on with receptors, right, that could be shutting out and then not, using these products of the locus coeruleus, we don't know yet. Again, this is something that probably can be better answered by experimental models with more funding that we're starting to have now.
And the last one will be, what neuronal changes do you think is the in vivo LC imaging picking up? Volume decrement versus neuronal decline.
Let's see if I understood this question right, but I think I understood that the question is what is the substract of the signal that we see in neuroimaging. So
Yeah.
if I understood it right, there are different acquisition schemes proposed for locus coeruleus. Many of them is based on magnetization, so it has to do with iron accumulation. And the neuromelanin or lack of neuromelanin because it has iron. But there are some other acquisition schemes proposed that has to do with contrast and even some that has to do with connectome, looking at structures that are around the locus coeruleus. But this is an area of active research, and this is something that we think we are contributing with the radiologists, trying to understand what is the best imaging sequencing and why. So.
Well, I don't know if we have more time, but I would like to hear a little bit of the technique that you are using to take these images.
Oh, for the images? Yeah. Yes, sure. So as I said, the main problem in doing histology to imaging validation is that we need to have perfect registration. Mm-hmm. So the locus coeruleus is so small that if we are off by 2 millimeters, you're already too off, right? And 2 millimeters is even nothing in neuroimaging. So what we did is to try to make a combination of brain processing that will cause less deformation with very powerful computing tools that can deal with this nonlinear registration. So I can show very quick slides here because I think it's easy to show in person, the imaging. So one thing that we did, and it proved to be very useful, was to print models of skull of the same patient. So for everybody who got an MRI, especially post-mortem MRI, we had to do a CT to make sure there was no metal there. So we took this CT, created the 3D skull, and we printed in 3D. So instead of hanging the brain in a bucket and it get all deformed, we just put inside the skull over here. So now we have a collection because we have many brains. And also, instead of cutting the brain in small pieces that generate a lot of deformation, we can put in these very large blocks of psiloidin. And we cut it in these very large machines. They are not produced anymore, but we got one on eBay from 1910 that we use a lot. They had these very large knives. My kids, really, when they come, they are all impressed by this. So, you know, all techniques adapted to the new world that help a lot. And of course, the computing. We have to use supercomputers to deal with this data registration, because when you think on memory that you need to reconstruct MRI, you need about 10 megabytes of memory. But when you think about histology with all its resolution, we are talking about 80 gigabytes. So it's just 8,000 times more. So, you know, we have to combine this together. I hope it answered a little bit the question.
Yeah, yeah. Thank you very much, Leah. It was a wonderful presentation, and thank you for your time.
Thank you very much for your attention. We have postdoc positions open, so if you know anyone who would be interested in working with some of these projects, send our way. We'll be happy to review CVs. And enjoy the rest of your day and the weekend, and happy Halloween tomorrow.
Yeah.
Bye-bye.
Thank you, Liam.
Thank you.